Hello, my dear friends. How are you? I am Dr. Jasmeet from the chapter of carbohydrate chemistry. Today we will discuss polysaccharides. So, what are polysaccharides? They consist of repeating unit of monosaccharides. Mono means single unit, poly means many. They consist of repeating unit of monosaccharides, and how they are held? They are held by glycosidic bonds. So how they are useful? They are useful as storage form of energy and also have some structural functions. And they exist in two forms that can be linear, that is straight chain, and branch that contain branches also. So now we will come into two category of homopolysaccharides and second is heteropolysaccharides. Homo contains same units, hetero contains different units. So right, so homopolysaccharides are there. They consist of single type, right? Single type of monosaccharide. Homo means same and single type of monosaccharides. Now we will come to heteropolysaccharides. They are found by hydrolysis of a mixture of monosaccharide, right? That's why the name hetero. Now comes, now we'll discuss in this video, we'll discuss only homopolysaccharides with complete details. So the first homopolysaccharide we are going to discuss is starch. Starch, right? It is a carbohydrate reserve of plants and it is a homopolysaccharide, right? Homo means it contains the same unit. The same unit is alpha D glucose. And how the glucose units are held? They are held by alpha glycosidic bonds. So the starch has further divided into it has two components. One is amylose and the second is amylopectin. So now coming on to the sources from where we can get starch. First is vegetables, second is cereals, then roots and tubers, right? Now we'll come into the two components of starch. First is amylose and second is amylopectin. Amylose is 15 to 20% and amylopectin is 80 to 85%. So the amylose is water soluble and amylopectin is water insoluble, right? Now we'll come into the structure details of this amylose and amylopectin. So we'll start with amylose. Amylose is long and unbranched. It is straight chain and is unbranched, right? The second thing is that it has 200 to 1000 D glucose units and it is held by alpha 1,4 glycosidic bond in straight chain, right? Now the second component amylopectin, it is a branch chain, right? Straight chain contains 1,4 and branch is at alpha 1,6 glycosidic bonds and it looks like a branch tree. Then starch is hydrolyzed by amylase. Amylase, it exists in amylase saliva as well as pancreas to liberate dextrins, and dextrin will finally form disaccharide, that is maltose. Then maltose will finally degrade into form glucose, right? Amylose acts specifically on bonds alpha 1,4 glycosidic bonds which are present in starch. Now the detailed structure of amylose and amylopectin. So this is the structure of amylose. This is straight chain of glucose with 1,4 glycosidic bond. There is no branch. So this is amylose. Now suppose this is also a one amylase unit and this is one amylose unit. So branching is at alpha 1,6. So branching when added it will form amylopectin. So the difference is clear. Amylose is straight chain. Amylopectin contains branches at bonding 1,4 glycosidic bonds, right? Now we'll on further move to the second homopolysaccharide, that is dextrins. Dextrins, how they are formed? They are breakdown products of starch. So starch will further degrade to form dextrins. Dextrin will further degrade to form disaccharide, that is maltose, and maltose will finally form glucose, that is monosaccharide aldohexose. Now we are coming to the third homopolysaccharide, that is dextrins. They are glucose polymers, but they are produced by microbes, right? And they, how they are useful? They are useful as plasma volume expanders in transfusion, and they are also used in gel filtration chromatography. Now now we're coming to the fourth homopolysaccharide, that is glycogen. The other name of glycogen is animal starch, right? So it acts as carbohydrate reserve in animals. Now, where starch is stored, where glycogen is stored in the body, glycogen is present in liver, muscle, and brain, right? So it is also present in non-chlorophyll containing plants, and besides, it is also found in yeast and fungi, right? The structure of glycogen is similar to myelopectin. We have already discussed myelopectin. Myelopectin contains straight chain alpha 1,4 and branch at alpha 1,6. So the structure is similar. Straight chain alpha 1,4 bonding, and at branch chain there is 1,6 glycosidic bond alpha 1,6. Right. The number of glucose depending on the source from where glycogen is taken, for whether it is taken from liver, it is taken from muscle or brain. Right. Now we will discuss the fifth homopolysaccharide, that is cellulose. Cellulose is the most abundant organic substance in plant kingdom, right? And it is crucially found in plants and constant of plant cell wall. It contains beta 1,4 glycosidic bond. In earlier we discussed starch. It contains alpha 1,4 glycosidic bond. Then we discuss glycogen. It also contains alpha 1,4 glycosidic bond, and branch contains alpha 1,6. Now the difference between starch, glycogen, and glucose is and cellulose is that cellulose contains beta 1,4 glycosidic bonds. So it cannot be digested by amylase that break down alpha 1,4 glycosidic bonds. So hydrolysis of cellulose will finally yield cellobiose, that is a disaccharide, and disaccharide will finally form the monosaccharide, that is beta D glucose, right? And what is the importance of cellulose? It adds bulk to the faces, and it also helps in decreasing the absorption of cholesterol from the intestine. Now we'll come to the structure of cellulose. As discussed, cellulose contains beta 1,4 glycosidic bond. So we have two beta glucose. This is one glucose, beta D glucose, beta D glucose. So beta has OH group upside. So when water molecules leaves and bond is formed between beta 1,4, so the structure of starch is formed. It contains beta 1,4 glycosidic bonds. Now coming on to the inulin. So glucose was there. This glucose is aldohexose, fructose is a ketohexose. When glucose is polymerized to form a homopolysaccharide, we've got starch, we've got glycogen. Now, when fructose is polymerized to form homopolysaccharide, it is known as inulin. So it is a polymer of ketohexose, that is fructose. It is water soluble. Property number one, it is water soluble, and it has a low molecular weight, that is only 5,000. So now coming on to the sources of sources of inulin. It is found in daily herbs, garlic, and onions. Right. It is useful in assessment of GFR and kidney function test. Now comes chitin. It is a structured polysaccharide, and it consists of repeating units of N-acetyl D-glucosamine, and the bonding is beta 1,4 glycosidic bonds. So what are the where it is found? It is found in exoskeleton of invertebrates, especially insects and crustaceans. Now comes the bibliography from where I delivered these lectures. These are the books, and to now I would like to thank you, and please do like and subscribe my channel, and please press the bell button below. Thanks a lot. Best wishes.